And hello, friends. So we've been going through basic epidemiology, which I think everybody currently has a minor crash course in. That said, if you don't have at least a master's in epidemiology, I think you need to probably not be vigorously expressing your opinion about the pandemic in any way at all. If you have something to say about the pandemic and you don't have a degree in epidemiology, keep it to yourself. Or if you're interested in expressing an idea, no problem, about epidemiology, I can give you a number of wonderful schools that you can go get your Master's of Public Health in. Otherwise, you don't really know anything about the vaccine and about the disease progression, and maybe you need to just sit down. But far be it from me to bring politics into the discussion. Anyway, we're going to talk about sort of mechanisms, not including antibiotics and not including heartworm diseases and or antihelminthics or antivirals, but good old-fashioned immunity. And this, of course, is its own, very own upper division class. And we could spend an entire semester talking about immunity, but we won't. We're going to spend a week. Still, immunity is a critical part of health. And, people are constantly touting things, potions, lifestyles that are going to be immune boosters. And there are certainly some things that will begin to weaken your immune system. And, hopefully reducing those things will boost your immune system. I would say, though, your probably number one thing is adequate rest, good nutrition, less stress, maybe a little meditation. These are things that will help to increase your immune system. And having a really good immune system, let's face it, that is your number one defense against all of these things that we've been talking about. And an interesting thing to kind of begin considering is when we think about bacterial diseases particularly. Viruses, yeah, you catch those. But bacterial diseases, a bunch of bacterial diseases come from bacteria that are on you and in you all the time. Our lungs are filled with staph and pneumococcal bacteria. Our skin surface is covered in a whole range of bacteria. And these bacteria are either playing an important role in some kind of ancillary defense, mutualism, a parasitism of sorts, but then you have to keep them under appropriate constraint. And so no wonder when you become stressed, your risk of having various disease increases. You're seeing the effect of a reduced immune response. One of the classic experiment of looking at the role of the immune system has been seen in people that have acquired immune deficiency, that is, AIDS. The human immunodeficiency virus, because it attacks a component of the immune system that we'll be getting into in a moment, it is an experiment in which you can observe what happens if you don't have a functional immune system. There are a range of fairly rare mutations, these like the bubble boy and things, people that have mutations that generate or that cause them to have a very ineffective immune system. And their lives are dramatically changed. You have to be very, very careful about what you're being exposed to. The same with people that have HIV. Once their immune system begins to crash, they are now sensitive to all kinds of things that you and I take for granted, that we laugh off, that don't even bother us because of a reasonably healthy and functional immune system. So we have some immunity definitions and things. So resistance to pathogens, that is immunity, potentially. And it's a fairly lame definition, but the book's is even lamer. Regardless, we have two types of immunity. We can break the immune system kind of defense mechanisms into two different broad categories. We've got innate immunity, and these are things like physical barriers, like your skin. Things like the secretion of various kinds of chemicals, different kinds of enzymes. We've talked a little bit about the fact that tears and sweat will contain the enzyme lysozyme that'll break apart the cell wall of the bacteria. And so these are part of an innate system. They're not going to be affecting viruses. They're not going to be doing much on various kinds of eukaryotic parasites, but they do a fairly good job of keeping bacterial infections at a reasonable level. And so part of what we're talking about with innate immunity is just this general kinds of mechanisms that are keeping the bacteria at bay. So having a puncture in your skin is defeating one part of your innate immunity. Now, a more interesting kind of aspect of the immunity is adaptive, or also known as acquired immunity. And this is where we have immunity that is induced by and directed towards specific pathogens or foreign agents. This is where we end up with antibodies of the immune response. And so when we're generating an immune response to a vaccine, this is an injection. We also have you can immunize yourself with a whole range of different things. Some of them not even, pathogens. If you want to make antibodies against specific proteins, you can do it by just injecting yourself with that protein. One of the issues here is that we have to be careful that we don't begin making immune response to self. Autoimmune diseases are a real drag, and they do happen. So let's see what kinds of things we can get into. Innate immunity, to start with. We have the kind of obvious physical barriers. And these are things like our epidermis. And if you think about it, this is a fairly tough layer. You're not at risk of dissolving with soap. The epidermis is packed with intermediate filament proteins like keratin that are in fact cross-linked, and it makes it very difficult both to penetrate and it makes it fairly tough. So you don't have the risk of dissolving or tearing. And additionally, because this outer layer is constantly being sloughed off, the surface of the body is being sloughed off with the bacteria sitting on top of it. So, if you don't like this piece of skin, wait a while and you'll just get rid of it. So we have exfoliation. Yeah, be careful with that. Don't exfoliate too much. Internal epithelia. It can have multiple layers. So we've got, if you start digging into your stomach, you got to go a ways before you get into the abdominal cavity, thankfully. Most places are that way. Within that, we have viscous fluids and things that are going to be able to trap bacteria. We can also, like in the lungs and in a variety of different places, in the ears for that matter, we can move things around. So cilia can move mucus around. You can begin coughing stuff up. Oh boy. Muscles are able to move things. We've got urine flowing, saliva, sweat. You can move your GI contents. That sounds like a good time. Both ends, either throwing up or throwing out, or defecation. This is a way of ridding bacteria from the system. There's a whole range of internal hairs, like in the ears and nose, and these are serving as a filter to prevent things from getting further up. That's one of the things is that you've got a whole range of things that are keeping stuff from the deep parts of your lungs. And so, depending on how you want to stick things into your lungs, there are parts of the lungs that don't typically get exposed to a whole lot of stuff, and they don't really have great ways of getting things out. So the deep inhalation of bacteria-contaminated air, probably not a good idea. Sticking stuff deep into your ear, not great. I was just thinking about like sticking something way up your nose. No, let's don't do those things. So we've got then a pretty good physical barrier. In terms of the chemicals, we're talking about all kinds of things that are part of this, but just as a notion, the fact that we have relatively high salt concentration on a range of our bodies and our skin, this is going to serve as an osmotic stressor for bacteria on the skin. Fatty acids, these are going to be able to partially dissolve cell membranes. We also have enzymes that can actually begin breaking down bacteria. So lysozymes are able to break down the bacterial cell wall. We've got a whole range of proteases in the stomach and intestinal enzymes that are going to break things down, call them food at that point. Acids, we don't typically have a whole lot of bases, but acids in the skin, stomach, the vagina, these are lower pH areas that again are making it so bacteria aren't able to grow as well. And certainly we're talking about the more generic bacteria. There's always going to be guys that can get in and that are optimized for that area, and that's how you get a stomach ulcer, because you've got Helicobacter pylori that is able to neutralize that acid in a little limited spot and actually get into you. So you had in anatomy and physiology some examples here of the different kinds of barriers. Looking at epithelial tissues, sort of histological, we've got the outer layer of dead skin, and then we have the inner layer of actual living cells that are constantly dividing, essentially stem cells, and cutting off cells here that then dry out. So when you're exfoliating, you're pulling this stuff off, and if you go too far, it starts to hurt. I actually have a splinter in my finger, and I'm looking forward to, after class, going up to my dissecting microscope, sterilizing my ultrafine forceps, and digging in my finger. And if you've never dug around in your body under a microscope, I encourage you to do so. You begin to see, first, how incredibly tough your skin is. It takes tweezers and a lot of pulling to begin tearing the skin off like that. It's fairly waxy at the very top, and then you start getting into a little bit juicier levels below, and you begin to notice that, when it starts to get juicy, that's when it really hurts. The top layer, you're not noticing at all. Fingernails are pretty interesting as well, kind of almost waxy in the way that they begin to peel up. And so the epidermis then has got, depending on where you are in the skin, a number of different structures like sweat glands and the sebaceous glands that are secreting oil. So this serves as a really good physical barrier. So internally, if you do have a cut, that ain't the end of it. We have other kinds of barriers here. And so we've got things that can remove essential elements from tissue fluids that make it so it's difficult for proteins or for bacteria to grow. We've got antimicrobial peptides, and these are very short proteins, and very often what they're doing is able to fit into the membrane and punch holes into the cell membrane. There's a whole set of proteins that are part of the immune system that are generically known as complement, and these are triggered in an immune and inflammatory response. And these are, again, things that can generically punch holes in cell membranes and cause the cells to lyse that way. Protein, or a class of hormones you have heard of potentially, interferon. These are produced by virally infected cells, and they are a signal to the neighboring cells that a virus has entered the perimeter here, and they can begin to affect the way cells will be taking up the virus potentially. So now we start talking about the immune system from a more cellular perspective. We've got a whole range of different kinds of cellular systems. And these are generic. They're not really specific to any one pathogen that we're going to see when we start talking about acquired immunity. We have the killer cells, natural killers, lymphocytes, and they can engulf cells that have been infected, dead cells that are essentially have committed suicide. They can begin and be engulfed, and essentially the components of that cell are hydrolyzed, and you can then dump them in different places in the lymphatic system. Phagocytes, this is a whole range of what we think of as white blood cells, or leukocytes, and they also are part of the inflammatory system. So a number of different kinds of categories of phagocytes, neutrophils, macrophages. These are some of the things we want to be thinking about, but the details are not overly critical at this point. So this class of protein known as a complement, these are proteins that are activated through bacterial infection. And we talked about bacteria infections with this idea of intoxications, where bacteria generate a toxin and release a toxin. And part of this has to do with the candy coating, or the outer layer of bacteria. Organisms have evolved to respond to this as, hey, this is something we need to be paying attention to if this stuff is inside us. There is a generic compound produced by bacteria called lipopolysaccharide, or LPS. And this is a long-chain sugar that has some fatty acids attached to it. And it is pretty much produced only by bacteria. And lipopolysaccharide is a really brilliant inflammatory agent. So you can inject LPS at a site, and you can mimic a lot of what happens when you have a bacterial infection. You'll start to get inflammation and the buildup of pus and all kinds of things just from LPS. So LPS and other kinds of molecules like that, they are activating the whole generic class of complement. And complement then is able to put in different kinds of pore proteins that can then actually cause holes to appear inside the bacterial cell. There are receptors on white blood cells, or phagocytes, that when they bind complement, they're able to interact with bacteria in this way. And so we can actually activate this whole system. You have a membrane attack, vasodilation can occur, and this is going to increase the flow of fluids to the area of inflammation. Lymphocytes are being recruited to this area, and then they are changing in the way that they begin to engulf a bunch of stuff too. Interferon is an interesting kind of system in that it is binding to a cell near a cell that has been infected with DNA, with a virus. And it is able to trigger the neighboring cell to begin generating antiviral proteins and begin blocking the uptake of the virus. Because remember, with viruses, if they don't get into a cell, they're not doing anything. And there are some intracellular bacteria, but not that many. Most bacteria are going to be forming biofilms or attacking cells and causing inflammation, but viruses have to get into the cell. So if you don't have the receptor for the virus, you can inhale, inject, eat as much of that virus as you want, and it's not going to do a thing to you. Whereas with a whole range of bacteria, the act of inhaling them, ingesting them, is going to cause inflammation, all kinds of problems. But the virus, if it doesn't infect the cell, it's not there. And interferon can be produced by infected cells, and in turn stimulating a whole set of antiviral genes in neighboring cells. Interferon is being produced when the cell is actually on its last legs, when the virus infection is so great that it's actually beginning to die. And it indicates to the neighboring cells that something is a little bit interesting happening here. So the infected cell dies, and its last gasping breath is to spread the word to other cells that something is awry. All right. Natural killer cells are another class of cells in which they very tightly bind their target cell, and then they release a range of molecules, granzyme that induces cell-directed suicide, and a delivery system, this protein complex known as perforin, which is perforating, that's a way of thinking about it, the invading host cell. And specifically, the enzymes are triggering this phenomenon known as apoptosis, apoptosis. And just as a general public service announcement, it is not pronounced apoptosis. If you come across somebody that says apoptosis, they are a bit of a backwards hit. Although, how backwards can they be? Because they're talking about advanced cell biology. But the second P is silent. Apoptosis. And apoptosis is known as cell suicide or cell-directed suicide. This is different than a cell just drying up and dying. That apoptosis happens through a cell triggering system, in that when a cell is directed to off itself, it's not just sort of randomly lysing, but it actually goes through a whole set of steps that are diagnostic of a cell in the apoptotic cycle. One of the first things it does is that the chromosome, rather than just kind of plopping out into the extracellular space, the chromosome is specifically broken up into small chunks. There are a whole set of proteins that are being induced during apoptosis. So this is not just a cell dying. This is a very specifically organized cell death. I mean, because the other way to think about this is that you can have cell death through just punching a hole and lysis. But this is an organized kind of stepwise destruction. A little bit like the difference between, your headquarters blowing up versus you're being invaded and you begin shredding documents and stepwise disconnecting all the computers rather than just abandoning the place and running. So here is a scanning electron micrograph of a macrophage right here beginning to engulf a bacterium. Go for it, dude. So macrophages are able to actually engulf and begin breaking down bacteria. And so this is a generalized scheme that is known as phagocytosis, in which you are essentially engulfing an area. There's all kinds of stuff that happen with phagocytosis. You can end up actually pulling water into cells, and so there are a lot of protists that carry this out as a way of drinking, essentially. And there are cells in humans that also, in animals, that can carry this as well. And they're pulling in the bacteria along with extracellular fluid and generating a membrane vesicle. This is actually where we have clathrin, a protein that coats the inside of the membrane and causes the membrane to actually get puckered in as a little bag and then cinched off. And so the endocytosis occurs, and we generate a vesicle that is known as a phagosome, which is going to become filled with all kinds of hydrolytic enzymes. And so there are another vesicle known as a lysosome, which contains all kinds of nasty proteases and lipases. It fuses, and then they go to work essentially destroying and turning into monomers, this little guy, until there's nothing left. And then those end products are being dumped back into the cytoplasm. So not only have you vanquished your enemy, you're also feasting on its bones. Lysosomes have all kinds of really nasty hydrolases and lipases and enzymes that are good at breaking stuff down. So we've got the initial chemotaxis, which is causing the phagocytes to migrate to the damaged site. And this is actually occurring because of wounds. And so you have a wounding that triggers inflammation. So if you ever wonder, you get a jab and it hurts. And after a time, it may start to get red. This is because chemicals have been released, hormones have been released from the damaged cells that trigger the inflammation response, the inflammatory response. And if it gets widespread enough, you start to run a little bit of a fever and start to feel achy. This is part of what happens when you have a wound. From there, the phagocytes can attach to different bacteria and material there and begin to actually ingest it by pulling it into the membrane. Once it's inside the cell, the lysosome dumps its nasty contents and begins digesting the cell inside the host cell. And then the debris is either going to be used by the cell, that is, we're going to consume the proteins and things and the fats, and that stuff we may not be able to consume, or it just doesn't get consumed. It can get dumped back out into the extracellular space, released by exocytosis. And now you're releasing parts of bacteria, another signaling system that says, hey, this guy is eating somebody, and maybe we need to start having even more phagocytes around that area. So it really is just a part of it, because once phagocytosis begins to happen, there are a whole set of chemicals that are being released that are triggering the next level of inflammatory response. So it begins with bacteria entering the body. So either through a wound or through inhalation, all these different portals of entry that we talked about. And depending on where it is, it can contact a phagocyte. Phagocytosis occurs, and because it occurs, that is triggering a whole range of different signaling molecules by the phagocytes. And these can activate the inflammatory process. You can begin secreting things that will kill bugs that are floating around out there. Depending, you might start to form a clot. And, this can even be separate from bacterial infection, but the action of the phagocytes is going to start this way. You can also have long-term regulation responses to the infection, and then the immediate killing of the bacteria. So phagocytosis is not just the killing of the microbe, but it also is the start of an alert signal that says something is happening. And this is actually part of developing the inflammatory response, which in turn is saying, hey, we've got a wound here. We need to begin not only affecting, or taking care of the invaders at this one spot, but recognizing there's a wound here and beginning wound process, healing, clotting, things like that. And so inflammation is another part of the innate response. And it's one of the things that you maybe have read. If you have a viral infection, should you actually be taking anti-inflammatories? They make you feel a bit better, but they may actually slow up the body's response to these different kinds of invaders. And so anti-inflammatories are working to inhibit these things. And what they're doing is increasing blood flow to the area, increasing fluid to the area. The increased blood in turn generates greater temperature, warmth, and you also begin to know it because it's painful. And so these are a whole set of chemicals that are being released by the damaged cells, the neighbors, phagocytosis, even the bacteria. So a little bit of injection of LPS with it too as well. Histamine, and you have taken perhaps an antihistamine. These are generating some changes in the vasodilation and permeability, so you got a lot more fluid coming in. Because what you want to do is sort of flood this area with all kinds of defensive agents, and by bringing more blood in and more fluid, you allow for these things to start taking place. Kymins are made in blood, and they're also vasodilators, but they also are serving as a chemotactic or a signaling system that's recruiting other stuff. Prostaglandins are released by the damaged cells, and these are actually part of the inflammatory system where we have enhanced vasodilation, permeability. Prostaglandins are inhibited by Tylenol. So when we start talking about pain relievers and anti-inflammatories, what we're doing is beginning to block some of these different kinds of systems. And so you may have read on the labels or hear the term of an NSAID, which is a non-steroidal anti-inflammatory drug. And Tylenol and ibuprofen, naproxen, these are all categorized as non-steroidal anti-inflammatory drugs. Another great anti-inflammatory is, of course, steroids. So if you've got a general inflammation going on, the doctor might actually prescribe a steroid shot. I think I was saying this in the molecular cell class, and sure, I'll go on videotape and saying this. I think that we can recognize that if a doctor is prescribing steroids, what they're basically saying is, you've got some kind of inflammatory problem. I don't know what the cause of it is, but here, let's take a course of steroids, and if that doesn't make you feel better, well, come back and see me. Because they are working as a broad anti-inflammatory system. Non-steroidal anti-inflammatories act at the cellular level, or in the case of Tylenol, they are inhibiting the release of prostaglandins. And so these can block the whole kind of inflammatory process. Leukotrienes are a set of hormone systems that are increasing the permeability. They're changing the way different cells attach. And so we start thinking about a blood clot can begin to form. And then of course we already talked about complement, which is just a general class of molecules that are released in the presence of bacteria. So we can think a little bit more about the inflammatory process in that we've got a puncture, ouch, and that little jabby thing is covered in bacteria. Probably a good idea why you want to use a sterile needle and you want to clean the skin surface. You're making a puncture, but at least it's going to be sterile. And so potentially, while that puncture is invading the skin, you're not inducing nearly as much inflammation as you would if, God forbid, you took a needle that had been used and covered in bacteria and punctured that in. Yeah. So as a general rule, let's use clean needles. Yeah, I can support that. And swab down the area. So we get bacteria that are in surrounding this jabby thing, and this in turn is going to signal the cells to begin releasing a variety of these chemical alarms. So here's what we're talking about. By changing the permeability of the capillaries, you've got now the ability of white blood cells, monocytes, neutrophils, to actually leave and move out of the capillary bed, and they can begin actually picking things up in this interstitial tissue here. And so, it's a thought of when we are injecting an RNA-based vaccine. It's got to get taken up by cells, and primarily it has to be taken up by red blood cells. Pretty much, you can, when you're giving a shot, you can do a couple of different kinds of ways. There is a subcutaneous shot, which when I think about this, and I wonder if you nursing people would know this. I don't know if you ever talk about administering something sub-Q, but when you do it in a dog or a cat or a rabbit, we actually would pick up a flap of skin and inject it like that. So it's not intramuscular, it's actually subcutaneous. Do you ever do a subcutaneous shot? Have you done one, or have you heard of it?
I've heard of it, but I haven't done one yet.
And I'm not sure what part of the human body you'd pull up to do a sub-Q shot. It was really easy in dogs and cats and bunnies that have floppy skin where you pull something up, and so you're injecting underneath the skin. When you're getting your typical vaccination, it is IM, intramuscular. And you can get one in your hind knee, you can get one in your arm, but they're jabbing in pretty far. And it may be that you do a sub-Q shot on people by just using a real shallow needle. Regardless, you have that puncture, and anyway, so by jabbing a needle into a dude, if you think about it, there's not a part of your body that you can jab a needle a quarter inch in and not hit blood because of the capillary beds. And so that eventually is going to get into the bloodstream. All right. So we're going to knock off here and talk about adaptive immunity on Wednesday. We should have our free vaccine clinic at this exact same time as well. Anyway, I'll see you on Wednesday.